Good morning. This is Mike Irwin, and it's a pleasure for me to introduce our speaker for Grand Rounds this morning, Dr. Stephen Cole. Steve is really an extraordinary scientist, and I've had the pleasure of working with him for over 20 years and have known him as a friend and colleague for over 30 years. His research is focused on the social environmental factors that influence health, and particularly looking at the alterations in inflammatory processes and immune system through transcriptional signaling. He's a truly interdisciplinary scientist. He's been trained in social psychology, human neuroimmunology, virology, and molecular biology. And he's really done such elegant and really creative work looking at how these psychological factors influence disease and the molecular mechanisms that explain those relationships. He is the founding director and really has charted a brand new field of social genomics, but he's a founding director of that laboratory here. And I would venture to say that he is the leading expert in the world on social genomics. And that's evidenced by a number of things. One, first of all, how productive he is as a scientist, publishing many articles throughout his scientific career in major journals. The kind of attention that his work has gotten from features in Science and in Nature, and also in more lay press, such as the New Yorker magazine. He's collaborated throughout the world. I mean, he is the go-to person for trying to understand how social and behavioral processes impact the genome. And he has, as he lists to me, over 200 research teams. And I know firsthand when I attended in a scientific meeting with him, hearing all the work that he's been doing with all the people that are presenting at that particular meeting. So he's certainly incredibly productive, not only in his own right, but also in developing collaborations. And finally, he's a friend and great colleague here in the Cousins Center, who, as I mentioned, I've known and worked with for 20 years. And he's going to give us, I'm sure, a whirlwind tour of his recent work and what he's been doing in the social genomics lab to interrogate these molecular processes. Steve, welcome.
Thank you very much. Let me just share up my slides here. So as Mike mentioned, I basically study the everyday life of the human genome, or how our psychological experiences, our social conditions, our cultural and economic and political context, how all of that world outside the body essentially gets inside the body and influences how the human genome operates. And often when I describe this kind of weird endeavor, people ask, where did you come up with this? Like, how did you ever start in on that kind of an expedition? So usually I've taken to starting talks by just kind of walking people through those kinds of experiences. So this is kind of a representative example of this, where these are data from a molecular epidemiologic study in which we're looking at one of the most toxic social risk factors known to epidemiology. In this molecular epidemiologic study, the rows in this little heat plot correspond to people in this study exposed to high versus low levels of this risk factor. Columns correspond to one of about 200 or so genes that are active at 30% or more differential rates in individuals exposed to high versus low levels of the risk factor. And the cell defined by the intersection of an individual row and a gene column, to the extent that that cell is colored red, that just means high activity of that gene and that person's white blood cells. To the extent that the cell is colored green, that means relatively low activity of that gene. So all these coherent blocks of red and green are really telling us is there's about 70 or so genes that are just systematically more active in the white blood cells of individuals exposed to high levels of the risk factor, about 130 genes systematically less active. And it turns out these aren't a random smattering of 200 genes, if you will, sort of drawn at random from the 20,000 genes in the human genome. They actually represent a couple of sort of coherent blocks, or you can think of them as kind of organized molecular conspiracies. So for example, prominent among the genes that are upregulated in people exposed to high levels of the risk factor are a block involved in inflammation, or the body's first line of defense against injury and the initiation of an immune response. Prominent among the genes that are less active in the white blood cells of individuals exposed to high levels of the risk factor are two blocks of genes involved in defending us against viral infections in general, as well as other kinds of extracellular pathogens. Type 1 interferons, key to antiviral responses, as we're reminded in the era of COVID, and immunoglobulin genes that are essentially sort of structuring the antibodies that we are now reminded play such a central role in defending us against infections. So this kind of, sort of, if you will, systematic shift in what the white blood cell is doing with its genome, I don't think would be terribly surprising if the kind of risk factor that we were looking at in this study were the kind of thing that we worry about most in epidemiology, a virus like SARS-2, or benzene in the drinking water, or cigarette smoke. But what interested me about this study was just simply the fact that the risk factor that was structuring these differences in gene expression was really just the extent to which people in the first few rows of the heat plot felt disconnected from the rest of humanity. So it was loneliness that was structuring these differences in gene expression. And that to me seemed like more of a provocative puzzle, if you will. It wasn't particularly clear to me why the human genome should have evolved in a way that would cause it to change its, sort of pattern of deployed molecular resources as a function of what kind of a social life we were having. So that, struck me as worth following up. And in actually what turned out to be fairly extensive series of follow-up studies, it became apparent that this coordinated shift in, if you will, sort of immunologic resource deployment coming off of the genome really wasn't unique to loneliness at all. In fact, this turned out to be a relatively generic, you can think of it as sort of danger reflex that is baked into the human genome. And over the last couple of decades, we've learned a fair amount about how this molecular profile gets activated at a, if you will, sort of mechanistic level. We know that much of this is mediated by the sympathetic division of the autonomic nervous system, where you have sympathetic nerves releasing norepinephrine and other mediators from sympathetic nerve terminals. And these end up signaling through typically beta adrenergic receptors into immune cells and systematically altering the gene regulatory signal transduction pathways that are active inside the cell, suppressing, for example, the transcription of genes that are involved in type 1 interferon, innate antiviral responses, and simultaneously stimulating the transcription of genes that are involved in inflammation, genes like IL-1 beta or TNF or interleukin 6. So this, if you will, wiring diagram, it turns out is sort of operating predominantly, at least in circulating white blood cells, in the context of a relatively small fraction of the total white blood cell pool, the myeloid lineage immune cells. So monocytes and dendritic cells in circulating blood, eventually differentiating into macrophages in tissue. These cells in blood comprise, somewhere on the order of 5 to 10% of white blood cells, but they seem to be disproportionately sensitive to this kind of sympathetic nervous system mediated signaling that's carrying information from our lived experience at the psychological level into changes in what's going on at the level of immune cells. And this happens not just for cells that are already present in the circulating, sort of white blood cell pool, but it can actually shape which cells are most prevalent in that white blood cell pool. One of the things that happens, in addition to advising existing white blood cells to change their gene expression deployments, if you will, is there's also a separate set of gene regulatory dynamics that are involved in actually creating those immune cells themselves. Immune cells, particularly these cells of the myeloid lineage, have a relatively short lifespan. So every day we need to regenerate billions of these cells. That's happening all the time in our bone marrows. And that, it turns out, is an ecosystem that is sensitive to this kind of social environmental influence via the autonomic nervous system. So we've discovered that people who are living in chronically threatening or uncertain circumstances, these brain-mediated perceptual and evaluative processes that end up structuring differences in sympathetic nervous system activity, both neural firing rates and to some extent the arborization of neural fibers throughout the body. One of the places that those nerve fibers terminate is in the bone marrow environment, in which the hematopoietic stem cells are busily regenerating the billions of monocytes and neutrophils and other kinds of myeloid lineages, immune cells that we lose every day. And what happens when these sympathetic nerve fibers become more active, this norepinephrine is delivered into that environment, that ends up essentially kicking off a cascade of signals that advises these hematopoietic stem cells to reduce their output of big, smart lymphoid lineage cells like B cells and T cells, and to really amp up the production of these myeloid lineage first line of defense cells. So monocytes and granulocytes get produced in the bone marrow, and then they leave the bone marrow and circulate through the blood and interact with other tissues throughout the body. Now, that by itself is not particularly damaging, but where things get rough is when those cells encounter an area of injury or damage in the body. What happens is, because those cells are more prevalent and because they're primed to produce a particular profile of immunologic response, they can essentially over-respond to certain kinds of diseases. So for example, let me just show you one example here. Sorry, I can't make my little panel go away. There it goes. Now I can see what I'm showing you. One thing that happens is when you get central nervous system tissue damage, these cells get recruited into that environment and produce hyperinflammatory responses that lead to neurodegeneration. Another example happens in coronary arteries in particular, where if you get damage to the blood vessel walls, the monocytes that get recruited into that damaged tissue will build atherosclerotic plaques more rapidly and more aggressively if you have more of these so-called classical monocytes being pushed out of the bone marrow in response to this stress biology. If these cells end up being recruited into your lungs and other airways, they can over-respond and produce pneumonia or tissue remodeling of the sort that occurs in asthma. These cells are also biased in the kinds of information that they deliver to other cells. So one of the crucial things for infectious diseases is for these myeloid lineage cells to collect information about what kinds of pathogens like viruses or bacteria are driving tissue damage, and then deliver that information back to the rest of the immune system in the context of lymphoid organs like lymph nodes. So one of the things that happens is these cells are primed to advise the immune system that what's going on is probably not a viral infection, but instead a bacterial infection. So they end up undermining optimal antiviral responses, particularly due to that deficit in type 1 interferon biology. And that ends up being a great thing if you're actually infected by bacteria, but it's a terrible thing if what's actually infecting you is a virus, because the immune system ends up sort of pivoting in the wrong direction and can kind of get behind the curve as a result of that. And then finally, these myeloid lineage cells get recruited into environments where tissue is being damaged, including by your own cells growing out of control. So for instance, in most solid tumors, when large numbers of monocytes get recruited into the tumor microenvironment and they don't find an exogenous pathogen driving the tissue damage there, they end up producing wound healing responses that inadvertently fuel the growth of the primary tumor and its metastasis into distant tissue sites. So in a wide variety of ways, these stress-induced sort of biases in immune system gene regulation end up promoting, in an almost like generic fertilizer sense, the growth of other kinds of diseases, particularly chronic diseases. What this system is doing, these days seems kind of counterproductive. But once upon a time, it probably made a fair amount of sense. Essentially, what this system is doing is it's taking an immune response that by default is very good against viruses, which is, kind of, as we are all reminded these days, the scourge of a social animal like humans. So by default, our immune systems are pretty strongly polarized for antiviral responses. But when these sort of fight or flight stress biology signals get sent into the immune system's vicinity, it essentially is programmed to reduce this antiviral bias so that it can increase its efficacy in clearing bacterial infections. So why would the immune system be doing that? Well, it turns out that once upon a time, there was a pretty decent correlation between these acute experiences of threat or danger and the types of pathogens that we were likely to encounter, or at least the immune system was likely to have to deal with. Essentially, back when stress biology was predominantly driven by things like this, and things like that are exceptionally likely to bite you or cause wounding injuries. The result of the wounding injury is typically a bacterial infection. You're not typically going to get a viral infection from these kinds of wounds. You're typically going to get a bacterial infection. So it's a great idea to use what's going on in the sympathetic nervous system as kind of a biological forecast about what type of pathogen you're likely to need to contend with if you're a cell of the immune system. Now, the obvious problem with this, which you're all aware of, is that in the modern world, the things that activate this system via our brain, the things that leave us feeling unsafe or insecure or outright threatened, generally aren't things that are going to physically wound us. They might kind of spiritually wound us, but they're not going to, sort of produce any kind of a injection of bacteria into the body where it shouldn't be. But we still have this, millions of years old wiring operating in our body that says if you feel stress or threat or uncertainty, if that ends up being the code delivered through the sympathetic nervous system, pivot towards this antibacterial pro-inflammatory response at the cost of antiviral responses. And that, especially if that happens chronically, day in and day out, if we lead a lifestyle of stress, threat, and uncertainty, that will essentially drive chronic production of these inflammatory molecules that end up functioning as essentially generic fertilizer for things like metastatic cancers, neurodegenerative diseases, and atherosclerotic plaques. So, in this context, you can kind of think of this evolved wiring connecting stress biology and threatening experiences to changes in gene regulation as sort of a recipe for how not to live, both in the figurative sense, in the sense that these sort of unpleasant lifestyles and life circumstances are what are driving this kind of molecular bias, but also in the very literal sense that if we run this molecular bias too long, it will end up driving the production of the chronic diseases that are the major factors structuring early mortality in contemporary social and ecological conditions. So you could sort of think of this as a, sort of a evolved program for converting misery into death, if you will, which is not, obviously the most adaptive thing to do. But this is as I mentioned earlier, a kind of a modern context-dependent phenomena. So, because it is so damaging in so many different ways to general health prospects, that led us to spend a fair amount of time thinking about what we could do to intervene to block these effects. So we started asking, essentially, more broadly, instead of how not to live, how should we be living instead? If we don't want to have this molecular machinery operating all the time, translating, threatening and insecure and miserable life circumstances into these kinds of health risks. So we started in, sort of asking a little bit about what kind of experience, socially, psychologically, or you could think of it in economic or political terms, across the whole spectrum of experiences that we could measure in these many studies that Mike mentioned we collaborate in. We started looking for signals about when we didn't see this threat-related gene expression profile, when we might have expected to, given the risky circumstances that a person was living in. And then we could ask, what was that person doing in a risky environment that somehow protected them from the adverse biological impact that this, sort of evolved wiring from experience to the genome is going to have. So this idea that you could think of it broadly as a question about how we should live. That's kind of not really historically been the provenance of molecular biologists. That's mostly been the provenance of, let's say, moral philosophers or their modern day descendants, happiness psychologists. And both the sort of historical philosophers and the contemporary happiness psychologists have really sort of tried to chop up this question about how people should live. What's the best way to be if you want to be healthy, happy, strong, and generative? They often sort of respond to that by distinguishing between at least two different versions of what philosophers have called the good life. The first version you can think of as sort of essentially Epicurus's recipe for the good life, which is to have a lot of good experiences, to see beautiful sights, eat tasty food, sort of have lots of fun, spend time with people you like. In that version, the good life is the sum of a bunch of good, happy moments, essentially. So you can think of this as the hedonic road to well-being. And that has traditionally been distinguished from this more Aristotelian concept of the best good life being one that is structured not so much by consummatory self-gratification in that Epicurean sense, but more through the pursuit of something virtuous, something noble, something self-transcendent beyond one's own immediate self-gratification. So this has historically been labeled sort of a more eudaimonic perspective on well-being, the kind of well-being that comes from doing something good for the world or for other people, pursuing something big in life, having a kind of a noble purpose or goal. So as we were starting this kind of work, we kind of sought to at least, sort of chop up sources of resilience, if you will, into these kind of two big blocks of well-being and ask whether both of them were similarly effective at kind of delivering us from that molecular soup of death that I just described there, or whether one might be more or less effective than the other. So we did a study where we collaborated with a positive psychologist who specializes in these kinds of analyses, and they would administer surveys asking people how often they felt happy or satisfied with their life as measures of hedonic well-being. Measures of eudaimonic well-being more had the characteristic of like, how often do you feel like your life has a sense of direction and meaning to it? How often do you feel like you're growing and becoming a better person? So you have a kind of flavor for these two distinct forms of well-being, or two different flavors of happiness or whatever. And when we went back to the molecular biology and then said, okay, where do we find, people with high levels of, let's say, that eudaimonic well-being sitting in, sort of gene expression space relative to people that have lower levels of eudaimonic well-being? What Aristotle, or admittedly, your grandmother would probably tell you this as well. People who had high levels of a sort of a sense of purpose or meaning in their life, that were, sort of deeply engaged in some really significant life mission, they looked great. They had relatively low levels of inflammatory biology, relatively high levels of antiviral biology. People who had high levels of hedonic well-being, essentially in the absence of low levels, in the absence of eudaimonic well-being. So they were happy, they had a lot of positive emotional states, but they were getting those positive emotional states not by necessarily doing great stuff for the world or making the world a better place, but more again by this kind of consummatory self-gratification approach. They frankly looked terrible, which was an interesting thing to us, because when we looked at their other metrics, other measures of well-being, they had very high rates of positive emotional states and claimed they had good social support and all kinds of other good things. But somehow, despite the fact that they felt every bit as happy as people with high levels of eudaimonic well-being and had similarly low rates of depression and anxiety and other kinds of affective indicators of ill-being, somehow the human genome had come up with some kind of split decision that wasn't tracking with that observation. And this has turned out to be a fairly reliable distinction. Somehow, somewhere in the nervous system, a memo is getting sent that says, sort of a happy life that's driven by virtue and the pursuit of things that are valuable to you and to the world in general, has a different kind of physiological fingerprint than the happy life that comes from just kind of feeding your own hedonic machinery. So since that time, we spent a fair amount of time, first, as you might imagine, that was kind of a surprise to us. So we spent a lot of time just replicating those initial results and trying it out in different contexts to see how reliable it was. It's proven fairly faithful. So it looks like there's a connection there. The next thing that we've done is tried to figure out where does that connection come from mechanistically? What's going on in the brain that would lead to these differences in peripheral neurobiology that are proximally regulating these immune cell gene expression profiles? And what kind of a protocol could we, sort of prescribe for a person to try and push more favorable biology at the level, at least, of these gene expression profiles? So really kind of thinking of eudaimonic engagement essentially as a strategy for reducing that conserved transcriptional response to adversity, a kind of a clunky term, I'll admit, but seemed to be a reasonably accurate rendition of what we were picking up earlier in terms of how stress biology ends up driving these differences in immune cell gene regulation. So I'll show you some examples of this. One thing, kind of as a big kind of roll-up conclusion, so far, we have not found that there is like some hidden eudaimonia nervous system buried in the body, that like somehow there are specific nerves that take purpose or meaning and deliver it out to white blood cells or spleens or things like that. What seems to be happening is that threat biology, which we've characterized with a fair amount of experimental rigor, so we know all the necessary and sufficient mechanisms involved for evoking these kinds of transcriptional responses, we can say pretty confidently that that stress biology is the proximal determinant of these threat-related gene expression signatures. So that led us to really kind of hone in on the conclusion, and I'll call this a soft conclusion at this point because there's still a lot that we need to learn about how eudaimonia operates in the brain and exactly what's going on there. But as far as we can tell at the moment, eudaimonia doesn't have a neurotransmitter that is somehow different from the sympathetic neurotransmitter norepinephrine or something like that, and is, sort of battling against it in the periphery. What seems to be happening is eudaimonic engagement is laterally inhibiting threat biology. And so when your brain feels focused essentially on something that you really regard as virtuous or valuable, that apparently reduces the outflow of threat biology through the sympathetic nervous system, and as a consequence down to these gene regulatory impacts in the immune system. It appears that a big chunk of this has to do with the arrangement of these two big systems in the brain, the wanting, seeking, hoping system, most characteristically associated with activity in the ventral striatum, these kind of dopamine-rich systems that are involved in the sort of valuation of things and the pursuit of things that you value, and this separate kind of more classically amygdalar system that is kind of structured around defending you against injury or threat. So what seems to be happening is when we get a lot of ventral striatal activity, and we've done this in a number of different ways, we can do a classic fMRI study and show people objects that are sort of valued or not valued, and then see how strongly their ventral striatum responds to that. And then we can ask, do differences in, let's say, the magnitude of responsiveness from the ventral striatum correlate with differences in these gene regulatory set points that seem to be kind of operating as a sort of a basal individual difference in the immune system. And here's one example of that sort of thing, where we're looking at people who are responding to images or primes that are sort of signifying values that they have already identified as particularly significant to them. So we're using this kind of semantic priming paradigm in an MRI scanner. This is work done in collaboration with Naomi Eisenberger and Julie Bauer. And you can see that the more a ventral striatum, if you will, sort of responds strongly to a value that a person has identified as significant to them personally, the lower their basal pro-inflammatory gene expression is. So this is one example of this observation. There seems to be some kind of connection between valuation in the brain and the downregulation of this threat biology in the peripheral nervous system that ends up sort of leaving significant fingerprints in immune gene regulation. So this connection has led us to kind of try to implement protocols that involve more of this pro-social or a eudaimonic engagement, and asking whether that results in causal changes in experimental settings in these gene regulatory set points in the immune system. So here's a couple of examples of this kind of thing. On the left, you can see results from a study where we had This is research done with Teresa Seaman in gerontology, and she's got a fabulous kind of community-based intervention program where they have older adults go into the public schools and serve essentially as teaching assistants, really spending a lot of time helping teachers with the kids that are, the small number of kids per classroom typically that are most at risk of kind of checking out or falling off of the educational, sort of treadmill and becoming, sort of like behavioral challenges for the classroom or, and sort of impairing their own outcomes. So when they have the older adults, sort of go into this program, there is a fairly reliable reduction in this threat-related gene expression profile. As another example, on the right panel, you can see an experiment done by another happiness psychologist where she randomized people to do kind acts for the world in general, or for specific other human beings, or for themselves. And as you can see, it's only people who were doing random acts of kindness for specific other human beings that really showed a favorable reduction in this threat-related gene expression profile. Doing good stuff for yourself, much to my personal disappointment, doesn't really seem to, sort of amp up your immune, sort of regulation in ways that would be beneficial. And neither does doing kind acts in the abstract for the world. There seems to be something about doing things that are directly meaningful to another human being that is kind of key to maximizing the impact of these kinds of effects. I'll show you another example of this that came up recently in the context of the pandemic. Of course, we all experienced the misery of loneliness and social isolation to some extent for months and stretching to years sometimes now. So as you might imagine, if you're a loneliness researcher and you're acquainted with the, sort of the health risks of those kinds of psychology, you really worry about, sort of, is this having adverse effects on people's antiviral resistance, for example? And if so, what can we do about it? It's not like we're saying, hey, go out and catch viruses. It's more sort of like, if you've got to restrict social contact, what are the ways that you can live subject to that restriction that are going to preserve your antiviral resistance most effectively? So we had a chance to look at this in a study that we were doing in collaboration with some primatologists. As you all know, monkeys, super sociable creatures. These are rhesus macaques living at the California National Primate Research Center at UC Davis. And these animals live in big half-acre field cages with typically like 50 to 100 other monkeys. So very, very socially enmeshed. And there's a well-established paradigm for looking at the effects of, sort of social isolation or loneliness, or you can think of the loss of social embeddedness, in which you take monkeys out of these big field cages and you put them into their own private monkey apartments. These are not particularly painful for the monkeys. They're, reasonable size. They get food, they get toys to play with. So they're not like horrible, torture chambers. But as you might imagine, if you take a monkey out of these highly sociable environments and you put them in essentially sort of their own monkey apartment, you see exactly what we saw in human beings: more output of these classical monocytes from the bone marrow and a knockdown in the activity of this innate antiviral system mediated by type 1 interferons. So not particularly a surprise to us. This is exactly what we've seen in studies of lonely human beings and what we discovered at that very beginning kind of anecdote I shared with you. What we were interested in, though, is an idea that primatologists have worked with for a long time, and that bears kind of a striking semantic resemblance, at least a distant one, to that first paradigm I was talking about, where you get older adults, essentially grandmas, to serve as teaching aids for young kids. What the primatologists had been doing for years is taking monkeys that had grown socially dysfunctional because of their, sort of long times of being isolated or growing up in isolation, and giving them what they call a therapist monkey. These are paradigms that are run by Steve Sumi and other, sort of people following up on the, sort of classic work of Harry Harlow, asking not just about the damage of social isolation, but what can be done to recover from that. And one of the few things that reliably works is to take a socially isolated, socially estranged monkey and give it a young infant monkey to take care of. So the therapist monkey really isn't a therapist in the sense that we're used to thinking about. It's somebody who's your sort of your peer or your advisor or your collaborator. It's really a monkey that just needs you, needs your help, and is kind of an appealing character to take care of. So this works for normalizing lots of forms of aberrant social behavior that evolve in isolated individuals. It turns out it has similarly good effects on immune cell gene regulation. We see instead of this bump up, when we put animals into these socially isolated conditions, but give them one companion monkey with, that completely blocks the bump up in classical monocyte production and the suppression of this antiviral response, even if there's relatively modest interaction between the animals. So just a small amount of highly pro-social caregiving seems to be enough to buffer the body against these isolation-related health risks. So that gets us to a bigger part of the story about eudaimonic well-being, valuation, and caregiving, which, in lots of sense, each of these domains has aspects that are not shared with the other domains. But it seems to be, from the kind of the integration of the results that we've gotten across these many, many collaborative studies, that it's really this Venn diagram overlap where, sort of what's most effective in regulating the immune system's gene expression profiles most favorably is this sort of world of valuation, ventral striatal activity, if you will, that is particularly structured around caregiving, caring, taking care of people, and especially intergenerational caregiving, taking care of younger people. Like I said before, that doesn't seem to be mediated, again, by eudaimonia nerves per se, but there is one wing of the sympathetic nerve, sorry, not the sympathetic, the autonomic nervous system that is relevant to this, and that's the parasympathetic nervous system, which developmental biologists and neuroscientists have increasingly in recent years implicated in caregiving behavior, both at the level of mammalian physiology necessary for the most fundamental aspects of caregiving in terms of nursing, as well as this kind of generalization of this fundamental sort of maternal-child bond, this redeployment of this capacity for bonding and caregiving beyond just your own progeny, to your, sort of spouse, to your peers, to your community, to sometimes very abstract entities that you can nevertheless feel deeply invested in and will be enough to kind of warm your ventral striatum as a result. So it's possible that some of this inhibition of peripheral stress-related physiology that's driving these gene expression differences is actually being delivered peripherally through the parasympathetic nervous system, which, as we all remember, laterally inhibits activity of the sympathetic nervous system. So it's not necessarily the case that the integration is happening solely within the central nervous system, within the brain. It's possible that we're getting, if you will, sort of competing signals, threat signals sent out through the sympathetics, caregiving signals, or valuation signals, or all is well signals, or I'm doing the right thing signals getting delivered through the parasympathetic nervous system and then laterally inhibiting in the periphery. So, and as a result, that's led folks to start looking more closely at, is there just a general relationship between parasympathetic nervous system activity and these gene expression profiles? And what we find as a particularly striking phenomena, interestingly, higher levels of parasympathetic activity aren't very reliably associated with lower levels of inflammatory gene regulation. But quite consistently now across several studies, we've seen that higher levels of parasympathetic activity are fairly consistently associated with increased antiviral gene expression. In other words, that suppression of the type 1 interferon system by sympathetic signaling through beta adrenergic receptors seems to be somewhat dissociable from the sort of the tranche of regulation that is headed towards inflammatory genes. And the social and caregiving quasi-parasympathetic, possibly implicating, central ventral striatal activity, something in that whole ball of neurobiology seems to be particularly permissive of more favorable gene regulation in the context of antiviral responses. So clearly a topic of great interest to us these days. So that's our sort of little tour of, sort of considerations about how we should live. I don't want to leave you the impression that eudaimonic interventions are the only thing that favorably impact these gene expression profiles. In people who are highly stressed, for instance, they've just been diagnosed with a life-threatening disease, trying to talk them into being, pro-social or generative is sometimes, sort of a tall order. This is particularly the case when people are at acute stress, like they've just been diagnosed with cancer. In situations like that, we think it's probably much more effective not to tell them not to be stressed, which just seems kind of futile at best and abusive at worst, but to really get them focused, basically to block that stress biology. Don't even try and talk them out of being stressed, but instead kind of head the stress biology off at the pass when it's trying to interact with the immune system. So beta blockers work great for this. Only, it turns out, non-selective beta blockers, because it's really the beta-2 adrenergic receptors that seem to be mediating most of these effects. But in some situations, sort of blocking stress pharmacologically, absolutely the right way, or at least the only viable way to go. Also, the kinds of wellness practices that Mike and his colleagues at the Cousin Center and many of you in the audience study, those definitely have reliable, small, but definitely consistently detectable positive effects on these gene regulatory profiles. And then there is a similar small but reliably detectable effect in, sort of these more eudaimonic, purpose valuation driven impacts on gene regulation. So this, as I'm wrapping up here, kind of gets us back to this question about why this is all happening. And it caused me to kind of think differently about stress. So I had kind of grown up thinking about stress in the sense that Walter Cannon originally specified this notion that the body has learned over millions of years of experience to change its physiology, apparently including its gene regulation in certain ways in response to our experiences of life. His whole paradigm for this was that this was fundamentally adaptive because it was self-protective. In other words, the body is wise when it's reacting to disturbance and danger in order to maintain the stability essential to life. So he sort of thought of all of physiology and evolution that drove it as a sort of a system for defending the self. What I've started to wonder, given these powerful roles of pro-social behavior, of sort of ventral striatal and parasympathetic biology and intervening in this, is I wonder whether that formulation is not just slightly off, and that whether what our physiology has really evolved to defend isn't ourselves per se, but what we care about. Many times what we care about most is ourselves, but not always. So I always wonder about that parent running into the burning building to rescue their child, or the person, sort of jumping into the frozen lake to help a complete stranger. These are situations that don't make much sense in terms of self-defense, but make a little bit more sense in terms of physiology fundamentally enabling the defense and advancement of the things that we care most about. So with that, let me just kind of wrap up with this idea that John Stuart Mill articulated. He, when thinking about the sources of happiness, felt that only those who have their minds fixed on some object other than their own happiness have it fixed on the happiness of others or the improvement of mankind. He really felt those were the only people who were truly happy. And I've come to wonder whether they might be the people who are most healthy as well. There does seem to be a connection between this pro-social engagement and this advancement of values that go beyond individuals and this kind of fundamentally healthy, sort of adaptive internal gene regulatory physiology. So with that, let me thank you for your attention. Let me also thank the many research teams that have collaborated in the studies that I've shared with you today, as well as the many sources of support for this research. And with that, let me pause my share here and take questions if we have time for it.
Absolutely, Steve. Thank you very much for that really enlightening presentation about social regulation of gene expression. We do have many questions that are listed here, and so without really any order of their particular area, I'm just going to go through some of the questions and see how far we can go. One of the first questions that was raised has to deal with the lack of bacteria in our lives and whether the overly clean kitchens and homes and so forth could be leading to an overreaction of gene expression in our bodies as well.
I think that's highly plausible. And in fact, this is one of the interesting kind of conditions that we contend with in social isolation research is that, again, as we've all been reminded in the last couple of years, togetherness is full of bacteria and viruses. You cannot separate that kind of social regulation at the microbial level from the rest of sociality, as much as we might try. It's just impossible. But one of the things we've started to question is whether we even want to separate that. We are an intrinsically social species, and for better or worse, we've bet, historically, all of our physiology, all of our adaptation and evolution is a bet that sociality, that the benefits of sociality will essentially outcompete the costs and burdens of sociality. That has served us well. We've taken over the world, and we're not that big, we're not that strong, we're not that fast, we don't have armor plating. Humans have done a great job by betting on the social option, but it comes with these complexities that include microbial consequences. And I think the question now is, like, have we bet so hard on sociality that by stripping out sociality, including microbial exposures, regardless, some coming from sociality, some just coming from living in clean worlds, are we kind of messing ourselves up by being too clean? So I think that's a great question, and I really wish we had definitive answers.
Thank you. The next question has to do with the hippocampus and specifically kind of dealing with how the brain can regulate the immune system. And the specific question is to what role the hippocampus has in T cell production and immune memory.
I would say, we don't know much of anything about that in human beings. There are hippocampal, sort of lesion studies and other kinds of experimental paradigms that are present in mice. But as a person who's been doing PNI, I can tell you, be cautious about mice. Within immunology, the maxim is, mice lie and monkeys exaggerate, basically. What happens in mice doesn't necessarily track what happens in the human immune system incredibly well, at least in all cases. So it's worth doing your initial research in mice to try and work this kind of stuff out, but you absolutely need to cross-validate those results in humans. And so far, we haven't had much success in connecting the hippocampus in humans to, sort of immune biology. So I wish I could tell you more than that. Again, this is also not terrifically the central area of my expertise in neuroimmune interactions, but my sense is that the kind of classic hippocampal paradigms usually are good probes for learning and memory and other stuff that is kind of stereotypically hippocampal. But there's probably a lot more the hippocampus does than just that. Like, we know, for example, it's absolutely essential for navigation, for just like spatial cognition in general. It probably does do important things in terms of the immune system, but we've had a hard time capturing that in the context of humans.
So much of what you've been talking about has to deal with the interactions between the immune system and social interactions. So the question in this case has to do with whether these relationships are applicable to people that may have clinical disorders that have impairments of social interactions, such as individuals with autism or autism spectrum disorders.
Yeah, so I would say the way we normally answer questions like this is it really depends. So the social world or the political world or the economic world, you could think of as a distal influence that is essentially getting into our heads. And one of the things that's being computed in our heads is, are we safe? Are we okay? Is the world an okay place? Or is it uncertain whether is everything okay? Or is it outright the case that things are not okay? So this kind of spectrum of confidence, you can think of it as generativity, like everything is good, no worries, go look for good stuff, do good things, to like this more kind of threat-driven self-preservation alarm system stuff. That, as far as we can tell, that connection is not different in people that have, let's say, sort of variant kinds of social sort of worlds or conditions like in autism. In other words, the connection from the brain through the sympathetic nervous system to the immune system seems to be still a bit there. Where the complexity comes up is that an autistic person's brain might perceive the world very differently than a non-autistic person. So they may have very different ecological conditions that lead them to feel personally threatened or insecure versus highly secure. And some theories of autism would suggest that actually, that where they would feel most secure is actually when they have very small, controllable amounts of social contact. And you don't want them sort of overwhelmed with social stimuli, which will make them feel, threatened and insecure. So the question really is the question about under what conditions is a person with autism, threatened versus secure. And that, if that's sort of, if you will, above my pay grade. That's more, obviously for the autism researchers to answer, and may well differ substantially across individuals, both across the spectrum in general, as well as, at any given position on the spectrum, people's personal history and, kind of the social, sort of affordances that they've been able to develop in their life probably modify that as well. So I think the fundamental wiring is there, and the connection to the immune biology is almost certainly there. But when it gets activated or triggered, I think is going to be very different.
The next question has to do, I think we have more time for a few more questions. So the next question has to do with how you might boost or alter or promote a eudaimonic response, particularly in seniors. And as a pet owner, this is a question that's newer to me, but there may be other processes that you'd like to talk about. The question has to do with what role might a pet have in creating eudaimonic experience in seniors? But more broadly, what other things that people might be able to do to promote that eudaimonic experience?
So, that is the $100,000 question. We spend a lot of time, part of the reason we collaborate so widely is just trying out almost everything we can think of that might potentially promote eudaimonic well-being, and then asking, does this have the impact that we want to have? And one of the things we're pretty kind of confident of at this point is that there is not going to be one right answer for everybody, and that in fact, most likely, different people will find the same protocol differentially rewarding, differentially engaging their caregiving systems or valuation systems or ventral striatum, or however you want to think about it. So a lot of what we do now, is sort of like, can we generate a menu of options for people to choose from as examples of the way they might optimize their personal level of eudaimonic well-being? And definitely pet ownership seems like a fabulous example of that kind of thing, just simply because it hits all kinds of anti-loneliness psychology, right? You've got a companion. It hits caregiving. I mean, pets generally are highly dependent upon their owners / temporary collaborators, whatever we want to call ourselves. And so, absolutely, I think it's possible. I think the question becomes, again, back to that story of like, what is the symbolic significance of this for you personally? There's another version of the pet owner, which is like, I got to go walk the dog. I got to feed the dog. In other words, if it becomes a burden to own a pet, then it's like obviously a very different exposure in the brain, a different experience, and that's going to kick off a different kind of biology that may be, more bereft of those favorable eudaimonic physiological consequences and may even be burdensome. So as long as you love your pet, which it sounds like you clearly do, it seems like it can't help but be a good thing.
Sort of not on the list of questions, but related to that, and we know that inflammation has such an important role in the regulation of behavior and social processes and can alter feelings of connection with others. Is there any evidence that alterations in inflammatory biology could actually boost or alter eudaimonic experiences? I mean, most of your research has talked about a eudaimonia is related to lower inflammation. But by the other angle, I'm particularly thinking about your studies with anti-inflammatories.
Right. Yeah, that's a great question. So at this point, the bulk of the sort of direct neuroscience studies is non-experimental, as you know. We know from these randomized controlled experiments that we can change eudaimonic engagement and see a consequent change in, sort of these kind of molecular measures of sort of reduced inflammatory biology and increased antiviral biology. So we know that there is a causal arc there. However, I suspect that you're right, and that there is a reciprocal causal arc from these kinds of eudaimonic engagements. From inflammatory biology to eudaimonic biology. Some of it probably taking place in the brain, although again, it's harder to map that out. But I think, in your own endotoxin studies, we see that sensitive, probably the most profoundly sensitive circuit in the brain to that kind of, sort of, you can call it fairly acute peripheral inflammation. Very, very significant effects there. So I think clearly that's the case. And I think what we're probably seeing is sort of like the flip side of what we saw with loneliness, where, clearly lonely behavior will get you this threat-related biology. And that threat-related biology, sort of feeds back onto the brain to make it more threat sensitive, more socially cynical, less rewarded. In other words, you get a vicious cycle where the biological consequences of adverse social conditions end up feeding back to undermine your ability to recover socially and to rebuild the kind of social fabric you need for optimal health. I suspect the same thing probably is true for eudaimonic well-being, that if you can favorably impact inflammatory mechanisms, that will take the boot off the neck of the ventral striatum, so to speak, and you'll be able to, sort of, in this kind of flywheel positive feedback sense, enhance the amount of time, the number of engagements that you have that are truly rewarding in this kind of ventral striatal sense.
I have two questions actually coming from the same person, but I think they're interrelated, so I'm going to ask them both together. One has to do with whether hedonics have a lower threshold of what they experience as threatening, so hence a higher level of threat gene expression, and then also how empathy in relationship to eudaimonics may serve as a mitigating factor.
Yeah, so to the first question, that's a great question. And I really wish we had a more satisfying set of answers. But at this point, what I would almost do is have to go consult with the positive psychology folks. I think they probably have done studies of that kind of thing where you take sort of classic amygdala threat biology, and you do kind of literally the psychophysics, like you work out, the gain functions, and you ask whether the gain function is stronger for people with high levels of hedonic well-being. My cursory understanding of the literature is that, sort of most positive psychologists think it's an offset, that they're not more sensitive to threat, they're actually less sensitive because they're just kind of dumb and happy. But I have to say, I am not a card-carrying member of the positive psychology establishment, so I just do not have enough, detailed knowledge of that literature. So I would suggest, asking people like, at UCLA, there's a good crowd of folks doing this kind of work. And certainly you could ask people like Sonia Lubomirsky at UC Riverside, or Barbara Fredrickson at University of North Carolina. They're the ones that I think are most likely to know whether someone's done that kind of psychophysical threshold question.
So I gotta jump in this last question because we're really running out of time, and this is really such a great question. I just can't let it go. Can you become eudaimonically depleted? Meaning, like, you're a caregiver in the hospital and you're caring for so many people that at some point, and these are unsuccessful eudaimonic experiences, and that you are actually driven towards hedonic experience, which can lead to depression and
yeah, absolutely. So there's no doubt about that. And again, we've all experienced that in the last couple of years. Some of us much more, intensely than others. But I mean, this is the whole literature on burnout. It's an incredible challenge. And one of the reasons we ask, how can you reignite this system, is because of the challenges of burnout. So that's a great question.
Thank you.
Thanks very much.
We could jump in with that last question. And I thank you very much, Steve, for this really great conversation and also presentation today.
Mm-hmm.